Hello and welcome to the microbial world. In this episode, we are going to be learning the review of biology of microorganisms of industrial importance. Also, do not forget to subscribe, like and send in your comment. Introduction: the microbial factory floor. Industrial microbiology focuses on the large-scale use of microorganisms to manufacture economically valuable products. This discipline bridges microbiology, chemistry, and engineering, supporting industries that produce medicines, food additives, biofuels, enzymes, and bioplastics. Industrial microbes serve as biochemical factories, converting low-cost substrates into high-value compounds through their unique metabolic and genetic capabilities. Defining and selecting industrial microbes. An industrial microbe is a microorganism optimized to transform inexpensive raw materials into valuable products efficiently. To qualify for industrial use, a strain must exhibit the following biological and operational characteristics: non-pathogenicity, safe for handling and consumption, for example Saccharomyces cerevisiae; rapid growth and high yield, capable of fast proliferation and high product output; metabolic stability, retains desired traits across generations; genetic manipulability, amenable to strain improvement through genetic engineering; ability to utilize cheap substrates, grows on waste materials such as molasses or whey, reducing production cost. Major groups of industrially important microorganisms. Group examples: major industrial roles: bacteria, Bacillus subtilis, Escherichia coli, Lactobacillus spp. Enzyme production, solvent formation, lactic acid fermentation, recombinant protein synthesis. Fungi, yeasts and molds: Saccharomyces cerevisiae, Aspergillus niger, Penicillium chrysogenum. Ethanol fermentation, citric acid production, antibiotic synthesis. Actinomycetes, Streptomyces spp, antibiotic and enzyme production. Algae and cyanobacteria, Chlorella, Spirulina. Metabolic powerhouses, products and pathways. Microorganisms are harnessed industrially for their remarkable metabolic diversity. Depending on the growth stage and environmental conditions, they produce different classes of metabolites and biomolecules. Primary metabolites, produced during the exponential log phase of growth and essential for cell survival. Industrial applications often involve overproduction of these natural compounds. Product: microorganism, industrial use, amino acids, Corynebacterium glutamicum. Food supplements, MSG, organic acids, Aspergillus niger. Beverages, plastics, preservatives, ethanol, Saccharomyces cerevisiae, alcoholic beverages. Secondary metabolites non-growth associated are industrially valuable. Product: microorganism, application, penicillin, Penicillium chrysogenum, antibiotic, streptomycin, tetracycline. Streptomyces spp, broad-spectrum antibiotics, pigments, toxins, Monoscus purpureus, food coloring, pharmaceuticals, biocatalysts, enzymes. Microbes produce enzymes, biological catalysts for various industrial applications. Enzyme, source, use, proteases, amylases. Bacillus subtilis, detergent, starch processing, pectinases, Aspergillus niger. Fruit juice clarification, lipases, Candida rugosa. Flavor enhancement, biodiesel production, bioconversion and bioremediation. Some microbes transform complex molecules into useful derivatives or degrade pollutants. Steroid biotransformation, Rhizopus nigricans modifies steroids for pharmaceuticals. Pollutant degradation, Pseudomonas putida and Alcanivorax species break down hydrocarbons and detoxify heavy metals. Growth dynamics and industrial process control. Industrial success depends on maintaining the optimal physiological state of microbes within a bioreactor, a controlled environment designed to maximize productivity, growth phases and production strategy. The typical microbial growth curve includes lag, log, stationary, and decline phases. Log phase, ideal for biomass-related products, for example single-cell protein, enzymes. Stationary phase, optimal for secondary metabolites, for example antibiotics. Types of fermentation systems. 1. Batch culture, all nutrients added at the start, product harvested at the end. Simple but discontinuous process, suitable for high-value products. 2. Continuous culture chemostat, continuous addition of fresh medium and removal of used broth, maintains microbes in a steady-state log phase. Ideal for bulk production ethanol, amino acids. 3. Fed batch culture, controlled addition of nutrients without removing culture. Maximizes yield and avoids substrate inhibition. The bioreactor environment. Large-scale bioreactors are fitted with systems for aeration and agitation, maintain oxygen and nutrient uniformity, pH and temperature control, ensure optimal enzyme function and cell activity. Dissolved oxygen dew monitoring, automated sensors maintain ideal oxygen levels. Sterility, prevent contamination to safeguard product quality. Genetic engineering and process optimization. Modern biotechnology enables the design of superproductive microbial strains through molecular modification and metabolic pathway optimization. Classical strain improvement involves random mutagenesis using UV light or chemical agents to induce genetic variation. Recombinant DNA technology and metabolic engineering, a targeted, rational approach to enhancing microbial productivity, blocking competing pathways, suppress genes responsible for unwanted byproducts. Overexpressing key enzymes, amplify rate-limiting enzymes to increase yield. Heterologous expression, introduce foreign genes, for example human insulin in E. coli or Saccharomyces. CRISPR-Cas systems, enable precise genome editing for metabolic optimization, downstream processing DSP. The final stage, recovering and purify the product, is often the costliest. Steps include cell separation, centrifugation or filtration to remove biomass, product release, cell lysis if product is intracellular, purification, chromatography, applications and economic importance. Microorganisms drive production in multiple industries.
Food, beverage, yogurt, cheese, wine, soy sauce, citric acid, pharmaceuticals, antibiotics, vitamins, vaccines, and hormones, agriculture, biofertilizers, rhizobium, azospirillum, biopesticides, Bacillus thuringiensis, energy environment, biofuels, biogas, bioremediation, bioleaching.